Hey everyone, welcome back to another video by Camroom 5. Now that's another biology video today where we will be discussing the structural adaptations of leaf. It belongs to the chapter of nutrition and transport in plants. When we look at the structure of a leaf, the superficial labeling would tell us that there is a petiole through which the leaf is attached with the twig. There is a big vein within the leaf which is known as the midrib. Then we got veins which are vascular bundles, and then the main green portion is known as the lamina or the blade. It's the broader portion. If we magnify the longitudinal section of a leaf, there's a whole different story that we're going to be discussing today. Like there's this upper epidermis, which is a set of clear cells. They do not have any chloroplast. They are clear so that light can easily pass through them. Then at the bottom we have the lower epidermis. The epidermal cells do not have chloroplast. Then we can still see that there are other different kinds of cells, like there's the cylindrical cells known as the palisade mesophyll cells, which are right below the upper epidermis and above the lower epidermis. Then we have the spongy mesophyll, which has irregular shape and they have wide air spaces. The palisade and the spongy mesophyll have both chloroplast in them. There are guard cells, which are kidney bean shaped cells, which make the pores known as the stomata. The guard cells also have chloroplast in them. When we further dissect the structure of a leaf, we can see that the main vascular bundle inside has xylem on the upper end, which is the xylem vessels that we have already talked about, and there are phloem cells, which are known as the phloem sieve tube elements. We will be talking about the phloem sieve tube elements soon when we'll be talking about the process of translocation. The xylem vessels are hollow, which means they are dead cells and they do not have any cellular content on the inside. So they are dead cells which provide more space. The phloem sieve tube elements are living cells on the contrary, so they have their own different mechanism. We will be talking about it. The open spaces you see are known as the air spaces, where you can have all these gaseous exchange happening between the mesophyll cells and the environment. On the upper end and on the lower end, there is a covering of a waxy cuticle. A waxy cuticle decreases the evaporation of water from the surface of the epidermal cells. Moving back to the epidermal cells, let's quickly write some stuff about them. Epidermis are clear cells, so they do not have any chloroplast. They do not have any chloroplast, which means they are not capable of performing any photosynthesis. They just allow light to pass through, which provides higher access of light for the underneath cells. They also decrease the loss of water by transpiration, plus they protect the leaf. Let's talk about these cylindrical cells, which are known as the palisade mesophyll cells. These cells have a cylindrical shape. They are arranged in rows right after the epidermis. They have chloroplast, which means they can perform photosynthesis. They are close to epidermis for a reason, because they have more chloroplast. They have the responsibility of photosynthesis, so they are close to epidermis for access to light, so that they get light directly. And they can perform obviously more photosynthesis through their more chloroplast present. Let's talk about these spongy mesophyll cells. They're called spongy for a reason, because they have a lot of air spaces within. They're irregular shaped, so they do not have a fixed shape like the palisade. They do not have it. They also contain chloroplast, which means they are also capable of performing photosynthesis. They have wide air spaces between these cells for gaseous exchange. So all these gases like carbon dioxide, oxygen, water vapors, they can be exchanged between the cells and environment through air spaces. Let's talk about the very important guard cells. The guard cells are kidney bean shaped cells. These cells are very important for transpiration. So they are kidney bean shaped. Another thing I want you to notice is already drawn in the diagram. They contain chloroplast, which means they can perform photosynthesis. And another important thing is that their center, let me use a thinner pen. The center, the opening, is known as stomata. These pores are openings through which the gases can be exchanged with the environment. By the way, you can see that their cell walls are thicker near the edges, and then their cell walls are thinner near the side facing stomata. Why do we have unequal thickness of cell walls? That is for a reason. What happens is that when these cells absorb a lot of water, they become turgid. When they become turgid, they try to expand. But they can expand only sideways, like only horizontally, never vertically. That is because of the unequal cell wall thickness. And that is how the stomata is open more wider, in the sideways direction, like horizontally. Let's quickly talk about the star of the video, which is the vascular bundles. The vascular bundle means two things. It means the xylem vessels and it means the phloem. Xylem or xylem vessels, let me write it, these are dead cells. We have already talked about it, but phloem cells are live. Xylem has lignified cell walls which provides more support, and also that is the reason for the death of the xylem cells. Phloem does not have any lignified walls. These are living cells with cellular contents. Xylem transports water and mineral ions towards the leaf, while phloem, it transports the assimilates. Assimilate means the final food product. So they transport amino acids and sucrose from the leaf mainly towards the sink region. Now, while we are here at this leaf structure, let's talk about photosynthesis again, and let's quickly revise it. For photosynthesis, we need light, which is obviously passing through the epidermis and reaching our palisade and spongy mesophyll cells. What else do we need? We need water, which is coming to the leaf from the xylem vessels. Remember, the roots transported the water towards the xylem, and finally the xylem in the leaf structure is giving this water towards these cells. It is all happening through osmosis. In xylem, we don't have osmosis, but from one leaf cell to another leaf cell, that's osmosis. So osmosis means movement of water from higher water potential area to lower water potential area. Another important thing happening here is to keep a track of carbon dioxide. Let me use a different colored arrow. Carbon dioxide comes from the environment through the stomata by the process of diffusion. So the environment has higher carbon dioxide concentration and the gas air spaces have lesser. So carbon dioxide moves in by diffusion and then it keeps on moving from the air spaces towards individual cells. Like, let me magnify this cell and you can see that a single cell gets light, water and carbon dioxide, and through the presence of chlorophyll inside the chloroplast. 6 moles of carbon dioxide combined with 6 moles of water to give you a glucose molecule and 6 moles of oxygen. This process is not very simple, but for O levels that is okay. So carbon dioxide comes from diffusion by the stomata, water comes through xylem using osmosis, light energy is available, and then finally you get glucose, which is our sugar product, and oxygen, which is our byproduct. Oxygen goes out during the daytime. Now let's quickly talk about transpiration also, which is a very interesting topic. Remember, xylem was providing water to all the cells by osmosis? Now let's have a closer look. All these cells, when they get water accumulates on the surface of cell wall in the form of a film. and through the heat which is available from the environment, water begins to evaporate from the surface of the mesophyll cells. So you can see water is evaporating from the surface of the cells using the heat from the environment. These water vapors from the air spaces go outside using the stomata. So step 1 is that there's a film of water on the surface of mesophyll cells. Step 2 is that evaporation happens on the surface of the mesophyll cells. The vapors are then collected inside the air spaces. And there are more water vapors inside the leaf than outside, so there's a diffusion gradient. And all these water vapors then start moving outside through the stomata. So evaporation happens first and then diffusion happens. That is why you can also see the phrase evapotranspiration, because evaporation happens on the mesophyll cells level and then diffusion happens from the air spaces outside the stomata, and then water vapors go outside.